the arm, and I like to refer to it as the claw sometimes because it kind of looks that way, and then the stem. We can also refer to this as the FC region, and this up here as the FAB region. The FAB region is composed of two parts, a heavy chain and a light chain, and it is here. I'll go ahead and put the other chain in here. It is at this area here that you'll find the specificity for the antibodies. So just like the B cells that we were talking about and how they have a very specific surface receptor that binds to an antibody, that antibody that they produce has that same receptor so that it can bind to that same antigen. So all of the antibodies that a particular B cell will release will all have the same receptor, same surface receptor, if you will. So that means then if this is our bacterium, our antibodies are going to bind, if you imagine these are on the surface of our cell, our antibodies will bind to those molecules like that. So now you've got your bacteria cell becoming bound by antibodies. In this case, we would say this bacteria cell is becoming opsonicized. So this is the specificity for the antibody. Right there in the claw region, that's where it's going to recognize the molecules of the bacteria cell. Down here at the FC region, we have also a functional part, an active part. But this region here gets recognized by the macrophage. So our macrophage can recognize the FC region and then engulf this bacterium. You understand the beauty of that? Now the macrophage doesn't have to have surface receptors for each and every potential antigen that it can come in contact with, or for each and every different antibody that might be out there. No, our macrophage only has to have surface receptors for the FC region. And wouldn't you know it, the FC region of antibodies is pretty consistent. It's conserved. It's pretty much the same in most antibodies. There might be some subtle changes from one class to another class, but then our macrophage only has to recognize a few different FC regions, and they don't have to recognize 100 million different FAB regions. A couple of things that we can say about our antibodies with regards to their function. They can neutralize bacteria or toxins just by binding to them. They can immobilize bacteria simply by binding to them, binding to their flagella, causing them to bunch up. They can agglutinate, sort of the same thing, binds to them, causes them to bunch up. Opsonicize, like we've seen up here. Cause complement activation, and cause or involve in antibody-dependent cellular cytotoxicity. Okay, this is what I mentioned last time. This is the idea that certain cells and invaders and things cannot be engulfed. And so if we attach antibodies to them, they can be flagged for the cytotoxic cells that can come along and destroy them. And so that's what will happen here with antibody-dependent cellular cytotoxicity. Big worms and things can be bound, and then once the cell that comes finds those worms, the granulocyte that will do that, the eosinophils involved in antibody-dependent cellular cytotoxicity. Didn't say much about I haven't said much about the lymphatic system, and I don't really want to spend too much time about it. Not that it's not important, it certainly is. It's where we are going to find the majority of our white blood cells. It's where you're going to find most of the T cells and B cells. The lymphatic system is a one-way vessel system that essentially collects excess tissue fluid and waste and garbage and stuff and delivers it back to the circulatory system. In our lymphatic system, we have lymph nodes, and these lymph nodes are staged throughout the vessel system, and they are jam-packed with B cells and T cells. So all of the material that are in your tissues that's considered waste and bacterial products and stuff like that, all of that makes it into the lymphatic system. Okay, so that means that the B cells and T cells that you have jam-packed in your lymphatic system is going to be exposed to all of that stuff. It's a much better system if you think about it. It improves the chances of your B cells and T cells becoming exposed to the antigen. Rather than them having to circulate into the system and come into the tissues and find these antigens, they can sit into the lymph nodes and let the bacterial products come to them. So essentially they'll get washed over with the bacterial products, and hopefully they recognize one and bind to it. In addition to the lymph nodes, there are a number of organs that make up the lymphatic system. The one that you might be familiar with, the spleen. The tonsils are also considered part of this. The appendix can serve as part of the lymphatic system. Peyer's patches. Any place where you have a high concentration of white blood cells. And that's really why we're, each of those places we mentioned, find high concentrations of white blood cells. Okay, so let's get back to our exposure here to an antigen that was presented to a T cell. T cell recognized it and caused clonal expansion only after it had been presented that same antigen by a dendritic cell. Okay, so if we understand what's going on here, before we get to the point where we've got major antibody production going on, and so our bloodstream is flooded with antibodies, we've had to first have our dendritic cell come in contact with the antigen. Then our dendritic cell has to move into the lymphatic system and come in contact with the T cell. Then present it to the T cell. Then the T cell has to leave the lymphatic system and come in contact with the B cell that is also having come in contact with the antigen and is presenting it to the T cell. It takes a while for all that to happen. In fact, upon first exposure to an antigen, it can take up to 10 to 14 days before you start to see antibody production to the level that would be sufficient enough to fight off the infection. That's why these guys are so great. The memory B cells don't have to go through all this rigmarole. Memory B cells, in a sense, have already been activated. So if a memory B cell comes in contact with the antigen again, then we start undergoing clonal expansion, production of plasma cells, and then antibodies right away. There is no delay. There's no 10-day delay. We have antibody level increase in a couple of days as opposed to 10 days. So you can fight off the infection, actually prevent it from reestablishing a second time. That's why the memory cells work so well. They don't have to wait, you don't have this 10-day delay, if you will. Because during that 10 days, the microbes are going to be replicating, and that's pretty much you're going to be relying on your innate immune system. You're going to be relying on the macrophages and the neutrophils at that point. And then once the antibodies level increases, then we start to stop the infection, recover from it. I should mention that there are a few antigens that don't require the T cell to activate the B cell. They're T independent, and in most cases we're talking about lipids and carbohydrates where there are many repeating units. And so sometimes some B cells, imagine this is a B cell, if you have lots of those repeating units now binding like you might in a car so that they all get bound up, this could be enough to stimulate the B cell to undergo clonal expansion. So these are T independent. You don't need the T cell, but it's almost always carbs or lipids that will be T independent. Protein pretty much requires presentation. Okay, let's turn our attention briefly to the T cells. So the T cell that I've been talking about for the most part is the T helper cell, or the helper T cell, depending on the book that you're reading. And this cell pretty much is involved in regulation. Regulates, as we've seen here, the immune response. You know, regulates the B cells, regulates macrophages as well. There are, with regards to, let me finish this thought here. We also have cytotoxic T cells. And as the name implies, they're destroyers of cells that have been infected. Couple of things to say about T cells. Again, they're going to recognize very specific molecules, so there's high specificity here. They never produce antibodies. Only B cells produce antibodies. T cells never produce antibodies, and they never interact with free antigens. Okay? The T cell must be presented the antigen. It can't just bump into a free microbe roaming around and devour it. It won't work. It's got to be presented with the antigen first. 3 antigen-presenting cells, right? The B cells are antigen-presenting, the macrophages are antigen-presenting, and the dendritic cells are antigen-presenting. Those are the only 3 cells that present antigens. When we look at the surface of the T cells, we notice that on T helper cells, they have a surface protein called a CD4 protein, and cytotoxic T cells have a surface protein CD8. I bring this up because the CD4 surface protein is what the AIDS virus has recognized. So the AIDS virus infects T helper cells, but it does not infect cytotoxic T cells. We talk about their functions again. Mentioned that the T cells are going to judge the significance of the antigen being presented. The cytotoxic T cell, it induces apoptosis. I think I saw a video where they pronounce that apoptosis. Apoptosis. This is cell death from the inside. So if you think about this, if you're infected by a bacterium, you don't really, maybe it's producing toxins or something like that. You really don't want to lyse the cell and have it explode and release all of that stuff into your circulatory system or whatever. So instead, apoptosis is a destruction of the cell from the inside, so it kind of implodes on itself. That way we don't release all of the negative stuff into the environment. Our T helper cells, they release a whole host of different cytokines. Some of those cause the proliferation of B cells, some of them cause the proliferation of T cells. So some of the cytokines the T cells release can feed back to the T cell and cause the T cell to reproduce. They can stimulate natural killer cells, they can increase monocyte production, they can change endothelial cells so that it allows fluids to move across.
What did you say they stimulate? B cells? They stimulate B cells?
Yeah, just like the clonal expansion, they can do that to themselves as well. One other thing that we want to mention here with regards to these guys is that they recognize MHC molecules. Both T helper cells and T cytotoxic cells are recognizing MHC molecules. What are MHC molecules? Major histocompatibility complex molecules. These are molecules in which the antigen is